Okay. Plan for this afternoon, just sort of picking up where we had got to. We had started the circulatory system and we'd done blood, but not quite all of it. I wanted to do a little bit more on blood groups, but not very much because it's not terribly significant, but it gives us a link to the immune system. What we've got left to do in this unit, there isn't an awful lot actually. And not to scare you too much, but your exam is 4 weeks tomorrow. All right, so yeah, you should have a revision plan underway and should be starting to sort of go over stuff. I will give you some guidance on what to revise, but yeah, because so much of it was done before Christmas, you need to be starting to sort of get that under control, and that's on top of the assignment, of course. The plan sort of pretty much goes as follows. So this week I think we finished the circulatory system this week. So I'm going to do the heart this afternoon, which doesn't actually take very long, which is a bit of a pity because actually it's my favourite organ. I like doing the heart. Tomorrow morning we'll then do blood vessels, the whole circulation, a little bit of practical stuff on things like blood pressure and that sort of thing if you want to take blood pressure. But also then a quick sort of revisit of the assignment because I will also look at control systems tomorrow. So things like, what controls your heart rate and your blood pressure. Then we'll sort of have a look at some questions as well. So we're starting to get into the habit of, responding to questions on the subject. That then just leaves us with the kidneys to do, the renal system. So that will be next week's job. And we then have a couple of weeks really just to get things like revision, get the assignment in and have that proofreading session. And revision and a mock exam and that sort of thing to get done before the exam on Wednesday. No, that's not right, is it? Is it on Wednesday? It is, isn't it? The Wednesday morning? Yeah, Wednesday the 6th of February is your exam. I've just realised I've put myself down for interviews in the afternoon. It's all right. That's completely the wrong way round. I was thinking it was a Tuesday. So we've got still sort of 1.5 systems to do, and we'll get stuck straight into that. And let's say we'll finish the circulatory one tomorrow, so we'll do a bit of tidying up there. So we had a look at blood structure, so the idea of plasma and blood cells, and you had a look down the microscopes to look at some blood samples. We had a quick visit of the assignment, obviously, without me telling you too much on that. Remember, there is a lot of help on the podcasts. If you haven't listened to those and you've made a start, you really should listen to those because they effectively take you through all the bits that you need to include. If you don't include the stuff on there, then, on your head be it. And if you include too much more, then, beware that you're not losing space for the bits that are on there. Obviously, a lot of the assignment revolves around using good illustrations as well. We looked at functions of blood in terms of transport, particularly we looked at oxygen transport with haemoglobin and this idea of the oxyhemoglobin dissociation curve or the oxygen hemoglobin association curve, that thing, and variations on that for myoglobin and fetal hemoglobin. We also looked at carbon dioxide transport, but that's not something that's it's not going to come up in the exam, so it was there just for completeness, really. And we then went on to the immune response, and I think that's all that there is on there. I didn't do anything else after that, did I? No. That was really just to sort of set the scene for the bits that you need to know from the PowerPoint on the immune system. And the other thing that's in there really is the types of immunity. So being able to distinguish between the active and passive responses. So, sorry, active is a response, passive is where you're just given immunity. and then the natural and artificial forms of it. Okay, so I'll just tag those on the end as being things that needed to be included. A horrible feeling the board's going to mess me about. Ooh, and there it is. Okay, that was the additional thing. If only I'd scrolled down to start with. And yeah, it is that one over on the far right there, the artificial passive example injection of antibodies. For example, antivenom doesn't give you permanent immunity, and it's not something you do yourself, but it's used for all sorts of other things as well. But it's immediate protection. It's things like the anti-tetanus that you have if you've stood on something nasty and that sort of thing. Right, now, associated with the immune system, I usually do just a little bit on blood groups, but we could spend ages on blood groups, and it's not something that has a particular sort of place in what we're doing. But blood groups is something that people have heard of, it's something you're familiar with. And the idea that blood transfusions have to be of the same blood type, you know, from the donor to the recipient, is down to the immune response. So just briefly pass through this. Now, I'm not sure if I've got that. You won't have it on your in your handouts. I don't think I haven't put anything on blood groups in there. But I might well have a PowerPoint on it, so I'm just going to check for that, just to give us a bit of a starting point. It would be on circulatory, wouldn't it? Blood grouping, that one will do me nicely. You haven't got this one, have you? No? Following the immune system, you haven't got one on blood groups? No. You've got the heart comes next, I think, after that. It probably is on Moodle. I'm just going to spend a little time on this. If I don't get through the whole heart thing today before my my voice gives up, then it doesn't matter too much. It can roll over into tomorrow. But we'll do a little bit on this first, because it reintroduces the idea of antigens and antibodies. And yeah, okay, let's have a look at this. There are loads and loads. I've put 30, I will have said over 30 systems that can be used for grouping blood. But the ones that we're most familiar with are the ABO system and the rhesus system. So if you know your blood group, which there's no reason why you should, then you'll have something out of the A, B's and O's. You'll be A, B, or O. And you'll be positive or negative, which is the rhesus system. So I'm just going to explain what those are in terms of antigens. The ABO system requires one set of antigens, or is determined by one set of antigens. And they come in 3 varieties, although that's not entirely true. If you have the O antigen on the surface of your red blood cells, you have this chain of 5 sugars up here, all right? Green, blue, red, blue, and then a yellow one branching off. And their names are over there on the side. So the green one is N-acetyl-galactosamine. The blue one is galactose. The red one's N-acetyl-glucosamine. And the yellow one is fucose. And if you've got type O, you have this chain of 4 with a branch. If you have type A antigens on your blood cells and nothing else, then you have the same as O, but with an additional N-acetyl-galactosamine. All right, so you've got that additional green one there to make it the A antigen. If you have group B, again, you've got the same as the O, but you've got an additional galactose, an additional blue one on there. All right, now, your actual blood group is determined by copies of a gene that you get from both parents, and you can have various combinations from your 2 parents. You could have 2, I'm going to use the word alleles. Alleles is the version of a gene that you have. If you've got 2 A alleles or 2 B alleles or 2 O alleles, pretty obviously you'll be A, B or O. If you get an A and an O, if you get an A from one parent and an O from the other, you'll be group A, because A is dominant over O. if you get a B from one parent and an O from the other, you'll be group B, because B is dominant over an O. All right? But if you get an A from one parent and a B from the other, you will be AB, because we describe those as being co-dominant. AB is the rarest blood group in this population in the UK, and O is the most common. All right, but O is the recessive. It's the weaker, if you like, of the alleles, and it's a good illustration of the fact that whether an allele is dominant or recessive has nothing to do with how many of them there are in the population. And I'll show you some maps a bit later on, which are quite interesting. Now, there is, in addition to this, there is an interesting blood group that doesn't even have the O antigen. Yeah, sorry? Yeah, the Bombay blood group doesn't have the O antigen. So, even somebody with group O is going to be a problem for someone with Bombay type, because these are the triggers for antibody production. Now, I've got lots and lots of slides that describe this. Here is the sort of illustration of them actually on the surface of red blood cells. So group O with its chain of 4, group A with its chain of 4 and an extra one, group B with its chain of 4 and a different extra one, and then group AB with both of the A and B antigens. In terms of the antigens that they have, then, if there is an A antigen present, they will respond to B antigens that enter the body, and that's an immune response. All right? And if you've got B antigens and nothing else, then you'll respond to A antigens that enter the body, because you haven't got them, therefore they're foreign to your body. If you've got A and B antigens, so if your group is AB, you won't react to any blood type out of these, because you can't, because you've got A, you've got B, you can't react to either of them. But if you've got group O, we can't put A or B into you, because you'll react to either of those and produce antibodies. And what the antibodies do, they don't clot the blood, but they make the cells clump together, which is a similar sort of process, and it blocks blood vessels and cuts off the blood supply to important organs. So this is why transfusions of the wrong blood type would be potentially lethal. So it's a really good illustration of this idea of antigens on the surface of cells and the production of antibodies against the ones that you haven't got, what we call the non-self antigens. We're not going through all that, but let's have a look at the maps, because they are quite interesting. Now, if we look at group O is where you've got an O allele from each parent, so you haven't got any A's, any B's. There's a significant difference in the occurrence of this across the surface of the Earth. I mean, these, I think, are fairly old maps, and with the movement of people all over the world, this could quite easily sort of change a little bit over time. But just generally speaking, and it looks at really our sort of evolutionary histories, and there will be, for some reason I don't know, because I'm not hugely interested in human evolution. I'm just not. There will have been advantages to certain blood groups in certain areas, but I don't know what. But if you have a look at this, the pale creamy colour is the lowest population percentage that's got the O blood group. I'm a bit confused slightly by this, because I would usually say that the percentage in our population is about 45%, but this is showing 60 to 70%. So I'm not entirely sure that it's showing the actual group O. It might be the O allele, but that's a bit harder to track. But even so, it gives us a difference. So we've got the lowest rates of O are in a very definite area there, in the creamy area. A lot of sort of splodginess elsewhere, but then it's really, really obvious, isn't it? South America is almost entirely O. So, quite a lot of the southern end of the North America. And a little bit there up at the top of Canada and Alaska there. We've got, some very distinct areas sort of coming in at almost like a band up that side, where we've got almost entirely group O. Right. That's O. If we Hang on, find another one. If we look at the A allele, now you find that in group A, and you also find it in group AB, of course. The common areas for this, well, the low areas, not surprisingly, where it was O, we haven't got a lot of A. So South America and the southern end of North America, hardly any A there. But we've got much higher bits of it elsewhere, and it's really interesting splodges. And Australia always strikes me as being interestingly stripy. So you've got, quite the relatively high proportion in the southern part of Australia, and then fewer people, more northerly you go. Then an interesting splodge across the top of sort of Alaska into Canada, but with a white splodge in the middle of it. Not quite sure how that's come about. And then, if you followed that across horizontally, you've got a bit at the bottom end of, what's that? Greenland. And then across a little bit further, that bit I'm never sure. Scandinavian countries, I think it's Norway up that side. But what turns into what? Which bit's Norway, Denmark, Sweden? I'm never quite sure. Yeah. So you've got, anyway, a bit at the north end of Scandinavia, anyway. So, and then an interesting sort of exclamation mark approach somewhere in Russia? If in doubt, that big bit over the top there, top right, Russia. So, yeah, very variable. This is the important thing I'm trying to get over. And then the B allele, that's really sort of distinctive, that hardly any B at all in the Americas generally. And then the highest stripe of it through the middle of, not quite sure what you call that, but top end of India. So you've then got, what do you have there? Pakistan. And then, up the middle. And again into Russia, I think, up at the top. The sort of highest concentration. But you can see, if that is an area of a particular population, then that makes sense that you've got one area of it. But it looks like it might have spread out from that area and never quite got to the Americas or Australia. And, it fascinates me as to sort of, how different the distribution of the different alleles can be. Anyhow, that's the ABO system. The other one is the D antigen. This is the one that determines your rhesus group, whether you're positive or negative. Now, the ABO antigen is a string of sugars, so it's a carbohydrate chain. The D antigen isn't a carbohydrate attached on the outside of the membrane. It's a polypeptide, which is a chain of amino acids, which is threaded back and forth through the membrane. So it's a different sort of substance. It's still an antigen. And we have, it's made of 417 amino acids. But this, at the left-hand end here, it says 2, NH2. NH2 is the amino group. So that's the amino group of one amino acid. And then the others are all joined together into a chain until you end up with the COOH group, which we know is a carboxyl group, don't we? So it's the acid group at the other end. So you would sort of read this chain from left to right, as it were. But you either have this or you don't. There's no variations on this one. And this was harder to find population data. So, I wouldn't be too sort of convinced by this particular set of figures. But the rhesus negative and rhesus positive, there are quite significant variations. So, you can see big differences in the proportions. But generally, there's quite a large proportion of people are rhesus positive. In our population, European population, let's say, it's roughly, I always used to think it was about 25% negative and about 75% positive. But like I say, the figures are tricky to find sort of accurately, but majority of people are positive. Now, the only time this really becomes significant, apart from blood donations and blood transfusions, you know, if you're receiving, then you've got to be receiving the right type. And you might have heard it in pregnant women needing to know if they were rhesus positive or negative. But the donation thing is important because if you are rhesus negative and you do not have that D antigen, if you put some positive blood in with that antigen, your body will attack it and make it clump together. So a negative person can't have positive blood. A positive person can have negative blood. And the blood that could be accepted by absolutely anybody would be that one at the end there, O negative. O negative has no A, no B, and no D antigens. Nothing to upset anybody. It's sort of plain blood, if you like. So if you watch any of the hospital dramas or anything like that, and you know you've got they come into casualty with an accident and lost a lot of blood, they start off with 2 units of O neg, don't they? So 2 units of O negative blood, and then 6 units cross-matched or something like that if they've lost a lot of blood, because you can't cross-match it immediately. So you start off by giving somebody plain blood, as it were. So if you are O negative and you're a blood donor, they will take every drop you can give them, because obviously that is the most useful blood there is. But the significant thing with this is an interesting little sort of sidetrack, and it affects babies. They call it hemolytic disease of the newborn. That says up there. Hemolytic means, well, lytic is splitting, isn't it? So it's blood destruction, basically. And it only occurs if you've got a rhesus negative mother carrying a rhesus positive baby. Okay? Now, a rhesus negative mother can't tolerate rhesus positive blood. And for the majority of her pregnancy, this is not an issue because mother and baby's blood do not meet. They can't, they're at different pressures, they might be completely different groups anyway. However, it's particularly the first delivery, or it could be a miscarriage or termination or anything like that, there will be mixing of the blood. Some of the baby's blood will get into the mother and vice versa, although that's not such an issue. So at birth, or whatever separation of the placenta there is, some of the baby's blood cells will get into the mother's circulation. And if she's negative and the baby's positive, she will produce an immune response. She will produce antibodies that will attack the baby's blood. Now that baby's fine. It's escaped. It's gone away. She can't affect it once it's left her body. However, if she then has another rhesus positive baby and is already sensitized, you've only got to get one blood cell sneaking across the placenta and she will produce an immune response that will destroy the baby's blood. So the mother will produce this immune response, so you get the anti-D antibodies that she'll make, but she also makes these memory cells that we met before. So it's like having an infection. So when the mother carries a second rhesus positive baby, she can destroy its blood, and this if it happens literally just before birth, a complete blood transfusion can solve it, but it would have been the cause of neonatal deaths, before this was fully understood. But what happens now is that rhesus negative women are given the anti-D injection. Anyone heard it? yeah, I have. So it's been around for some time actually, because my eldest son is now 34. So it's been around for a while before that. So I'd say it's probably been around for the best part of 40 years. The anti-D injection is literally just anti-D antibodies. It's like an antivenom. It's an artificial passive immunity. And so what happens is and actually they do it during pregnancy now, I think, just to keep down any odd cells that get into circulation. But they will give these antibodies within 72 hours of any delivery or termination, whether it's a miscarriage or anything like that, just to stop the immune response. Because then what's happening is inject the antibodies, they whoosh around the system, get rid of the antigens, and the body never actually knows they were there. Right.
29 weeks.
Oh, right, yeah.
But again, if you knock yourself in between or have a fall, you have to have another one.
Ah, right, yeah, that would figure, wouldn't it? It makes sense. Just
One negative, both one positive.
yeah, yeah, yeah. I mean, I've no idea what my sons are at all.
Find out for a negative mother.
Yeah.
They find out what the baby is straight away.
Oh, right.
They give them D's as well.
Yeah. Ah.
So
Yeah. I have no idea about mine, but I'm not exactly babies anymore. But it's a really good sort of application of it's a similar sort of situation to the anti-venom situation, isn't it? Or the tetanus, where you get immediate protection, but not long term. The antibodies are injected. They take out the antigens, and that stops the body from needing to produce any sort of response. And it doesn't create any memory cells or anything like that. So it just means that then, she's, the woman's body then has no knowledge of D antigens at all at that point. So, it's been very effective. At one time, when they first started doing this, they had to extract the antibodies from donated blood. And there was one bloke who lived in Glasgow, I think it was, who he gave blood several times a year, more than people normally would, just to have the antibodies. I'm not entirely sure why. Why he would have them, but he produced a large amount, and they extracted it from his blood and then put his blood back in, basically. I think he got some sort of award for having, donated so much of this. But I suspect they now make these injections because they used to be really expensive as well. So I remember being told when I had my first one that each injection cost something like £60, and we're talking 34 years ago. So I suspect that they've now got probably a lab system for actually creating them, because an antibody is only a protein. And you could actually produce them by genetically modifying bacteria or something like that. It'd be quite interesting to find out. But it's an interesting sort of application of the immune response that a lot of people have come in contact with. Okay, that I think is the end of the blood thing. So we'll park that and put that down the bottom. And we will move on to the heart. Now, you will find in your handouts that you've got diagrams of the heart, you've got a PowerPoint on the heart, and I'm going to sort of dip in and out the various bits of this. But I'm going to start off working on the board and doing a diagram. If I just load up my heart PowerPoint. And just a quick skim through it so we can see what's coming. Structure. We have some cardiac cycle stuff, animated stuff, and a bit on pulse points, sort of measurements. So that comes right at the end. So I may not need that today. I might leave that till tomorrow anyway. But what I want to do is to make sure that you understand the structure of the heart, and then the overall function in terms of, what's a heartbeat? What happens? So what we call the cardiac cycle. So in your handouts you have quite a lot of information. So you've got, you've got a sheet that's got two images on it, one which is an outside view of the heart and one that's an internal view. On the back of that, you've got an unlabelled heart, which is not necessarily something to do today, but can be used for revision. You've then got a sheet that looks a bit scary, but describes different changes in the heart during a heartbeat. And the chart on the other side is even worse, but actually when we get there, you'll see what that's showing. And then we've got a dominoes sheet, and then it goes on to blood vessels. All right, so the basic heart structure and function is where we're at today. Now, I'm just going to pause a minute and find some models. All right, let's have a look at structure then. I'm going to draw a heart and add a lot of labels to it. And you can either copy this or label the diagram that's in the handouts. It won't be exactly the same as this though. I'm going to make it a fairly straightforward version. And there are certain things that I want to identify on there, particularly 4 chambers, 4 valves, and 4 blood vessels. Okay. Now, the blood vessels thing, we're coming back to blood vessels tomorrow, so that will make more sense when we do that anyway. But those are the key points, and then a few other bits and pieces as we go. So I'm going to use the whole board panel with my drawing and then labels around the outside of it. So just bear with me because sometimes I have to redraw bits because they don't quite work out. Just a single one coming in there, which is not really right, but I'm going to do a double one coming in there. And then, this is where it gets a little bit tricky, is getting the, whoops, shouldn't overlap. Having to leave myself room for labels is always the problem. Okay. Yes, I have drawn these before. Oh no, I've not looped them over. Never mind, it doesn't matter. Sometimes I do, sometimes I don't. I'm just going to keep it side by side like that. You'll see in the diagrams and in the model that those blood vessels coming out of the top here, from the middle, actually cross over each other. But I've separated them into the 2 sides really to make it a little bit easier. Right, it's not one of my better diagrams, I will say. Let's just tidy the middle bit up here. I've still got something else I need to add as well. Oh no, board is just out enough to be awkward. Sorry, that's just going to make a bit of a tricky job out of that, isn't it? Now I've done it. Oh, heavens above. Do you know what? I'm going to have to start again. Sorry. Do apologise. It won't take me long. Oops. Okay. It's the bit down the middle, it's the hard bit. Still not going to cross them over. I keep it like that. That's better. Not a lot, but yeah, they come out slightly different shape every time I do them. I've got the valves are slightly misaligned, but this isn't going to be critical. They're very misaligned, actually. Now, one of the problems I have, it's partly because I'm female, I think, and partly because I'm a teacher, is my left and right. Now, you're looking at that diagram as though that's my heart. So this is the right, yeah? This is the left of that diagram, because that's my right hand, that's my left hand. The problem I have, apart from the fact that female brains are more inclined to mix them up, is that, of course, I'm talking to you, right, left, and then I turn to the board and it goes right, left. So I have to do this, and that is put... yeah. It would help if I put the right. You see what I mean? Straight away. It would help. You know, if by the end of my career I could get this done. That is the most bizarrely shaped heart I think I've ever drawn now I look at it. Never mind. 4 chambers. 2 top ones, 2 bottom ones. 2 upper, 2 lower. 4 valves. 1, 2, 3, 4. And 4 blood vessels. 1, 2, 3, 4. Okay? That's all I need out of this. All right? I really wanted to make it more even than that. That is quite peculiar looking heart. However, let's start by sort of identifying some important things. Now, the 2 sides are separate from each other. There is a wall between the 2. We call it the septum. Septum is a general name for a wall in biology, isn't it? Where else have we got one? In the nose, yeah. A dividing wall between the nostrils. It's a septum. So this wall between the 2 sides is a septum. And it separates the blood into oxygenated and deoxygenated. The purpose of that is to enable us to have a double circulation so that deoxygenated blood comes back from the body into one side and is pumped to the lungs, and then the oxygenated is kept separate, comes back into the other side and is pumped off back to the body. So it makes it very, very efficient in terms of delivering oxygenated blood around the body. And it's the right side that deals with the deoxygenated blood. Now, by convention, we use blue to represent deoxygenated blood. All right, so I've written blue, written in blue. And for the left side, it's going to be oxygenated, and the convention is that we use red. So I'm going to label the parts using those colours. So let's start with the chambers. Right side, we have an upper chamber called an atrium and a lower chamber called a ventricle. So, quite simply, we have a right atrium and a right ventricle. And then over the other side, we have a left atrium and a left ventricle. Okay, that's 4 out of those, 4 chambers done. Now, the way that the blood flows through the heart is that it comes in to the atria. So this vessel here and the upper branch and that vessel over that side are bringing blood in. And vessels that bring blood in to the heart are called veins. Right? The word vein ends in an IN. Consequently, veins go in to the heart. This particular one, it's called the vena cava, unfortunately. It's got a Latin name, which just means body vein. But there are 2 branches. There's a bit that comes from the lower part of the body, so that's the inferior vena cava, and a bit that comes from the upper part of the body, which is the superior vena cava. But I'm just going to put vena cava for now. That's bringing blood in from the body, whereas the one over this side is bringing blood in from the lungs. Now, the word that we use to refer to lung things is pulmonary, and so that is a pulmonary vein. So those are both vessels that are bringing blood in. I mean, we could actually show the blood coming in, but no. I think there's a bit of an issue with the line up on the board again. So it's 2 of our 4 vessels. The other vessels are the ones which start down here and then come out of the heart. So the one on this side is taking blood out that way, And that's going to the lungs. Now, because it's leaving the heart, it's an artery. Arteries away, so the A of arteries and the A of away. And we've got two branches because it's going to the two lungs, and I'm sort of slightly off that side. But this is the, I'm just going to label that bit, pulmonary artery. Okay, so you get, whoops, let's just try and correct it there. Pulmonary artery, so there would be a left and a right branch to the left and right lungs. And then this huge vessel coming from the left ventricle here is going to the body. It is an artery, but it has a Latin name, which is? It's the aorta. And that vessel arches over the top of the heart. You can see it on the models. And then it goes all the way down through the thorax, behind the windpipe and the esophagus, and into the abdomen. And you get branches going off it to all the organs. And one of the times you sometimes hear about it is if somebody's had an aortic aneurysm. And if it happens in the abdomen, they call it a triple A because it's an abdominal aortic aneurysm. Aortic aneurysms are incredibly dangerous. I mean, aneurysms are generally... an aneurysm is when you get a bulge in the wall of an artery. And it's a bit like, if you've ever seen on a tyre, on a car or on a bike perhaps, where the rubber goes thin and you're left with the sort of fabric part of it and it bulges. And that's what you get in an aneurysm, is that the wall of the blood vessel sort of gives way slightly and could burst at any moment. And if an aortic aneurysm bursts, basically all your blood just empties out internally, and it's a very quick way to go. If they leak for a bit, you're in with a chance of a repair, but otherwise, you know, they are, you know, very destructive. So we've got, what have I done with my blood flow here? We've got blood coming in from the body over this side, through the vena cava, into the right atrium. We haven't talked about how it gets to the right ventricle yet, but once it has, then it's pumped out through the pulmonary artery to the lungs. Then it comes back in on the left-hand side, through the pulmonary vein into the left atrium, then into the left ventricle, and then it will be pumped out through the aorta to the body. So I ought to put some other little arrows on there, because this is coming down here and then up there. So how it does that is the story of a heartbeat. And the same over this side with the deoxygenated. How does it go around a corner? Well, we've got to consider these other bits labelled on here, which are the valves. Two sets of valves. And I'm not sure I've got room for all the labels, to be honest, but we'll do our best. Well, no, let's do there are valves that separate the atria from the ventricles. So big sort of flaps of tissue between atrium and ventricle on each side. And what happens with those is the blood can go from the atrium into the ventricle, but if it tried to go back from the ventricle into the atrium, it would push the flaps of the valve together, so it can't get back. That's why it's a valve. Right? So it stops the return of blood into the upper chamber. The one on the right-hand side is made of 3 flaps of tissue, and it's called the tricuspid valve. So the tricuspid valve, not surprisingly, has 3 flaps. And on the other side, we have one that's made of 2 flaps. So that is the bicuspid valve, which also has another name. And interestingly, in medicine, it is often still called the mitral valve. Okay, here's the question I ask every year. What's a mitre? Who wears a mitre? Bishops wear a mitre. What is a bishop's mitre? No, no, no. Oops. Here we go. Smiley bishop. Always have smiley bishop. Well. Ah, hang on. Try again. That's a very strange shape. I'm having trouble drawing today. I'll always draw the mitre first. Bishop's mitre is his hat. It has those two pointy bits. Yeah? So that's why, that's the mitral valve. Two pointy bits coming together is a mitre. Are there any mitres in this room? Yes! Corner of your desks. They're mitre joints, aren't they? Yeah? Come on, you're all qualified carpenters, surely. But yeah, that mitre, you know, when you get 2 points coming together, is what the mitre is. It's a mitre joint. That's interesting. What did it do and why? On earth is that? Oh, okay, there's some very strange additional bits there. So yeah, that's a mitre. Okay, so it's an upside down mitre. The other two valves, well, one's at the bottom of the pulmonary artery, so we just call it a pulmonary valve. The other one's at the base of the aorta, so we call it the aortic valve. But they are sometimes called semi-lunar valves, and that's because of their shape. So what shape are they? Yeah, half moon. Yeah, semi-lunar. So on the right-hand side, we have this one, which is a pulmonary valve. And sometimes, of course, they need to replace valves, don't they? Because if a valve is leaking, then you're not necessarily getting the proper pressure behind the blood. And it can be sort of leaking back to where it's just come from. So, I mean, the mitral valve is a classic one that people have artificial valves put in, or pig valves. And it's quite a, you know, there was a series of about 3 television programmes, actually live operations, not so long ago. One of those was a heart valve replacement. And they, yeah, I seem to remember, it was a mitral valve replacement. And, you know, I mean, these used to be huge, big deals, but these days it's interesting, heart surgery is not the huge deal that it used to be. And, you know, when you look at it, it's amazing how much we know about it. But, I mean, you know, it is plumbing when all's said and done, isn't it? And it is a matter of pipes and valves. The one thing that really amazes me is that, you know, with all this knowledge and understanding of it, and the essential simplicity of this double pump, is that we haven't managed to find an artificial replacement. Not a reliable one. There are artificial hearts out there. So, you know, occasionally they will use them just to sort of see people through short periods of time. But there are not any reliable hearts. I think there are problems like, for example, you know, battery life. Because obviously, you know, because you can't sort of, oh, yeah, oh, my battery's running down a bit, I'll have to put it on charge. That's not going to work, is it? And then the other problem is the materials that are used, you know, because you can't have anything that's likely to cause a clot. So, you know, when you look at that and you think, actually, you know, when all's said and done, that is not a complicated structure. It's been really, really difficult to replicate. So, you know, I think that would be a, you know, quite a significant breakthrough in medical science. So, you know, if anyone, if you want to combine medical research with engineering or something like that, then, you know, artificial hearts, I think, you know, be cool for that. Right, what else to say about it? Well, that dividing wall, that's the septum. A few other points just to make about this. What's the heart made of? It is muscle. It is a very specific type of muscle. And it's the only place that you find it is in the heart. It's cardiac muscle. And cardiac muscle has a particular property. It's described as being myogenic. Now, all the other muscle in the body requires a nerve impulse to make it contract. Heart muscle doesn't. You could take a heart out of a body, keep it in the right conditions, and it will keep contracting, or even a piece of the muscle. It actually generates its own contraction. And it does it from one particular little area we're going to talk about in a minute when we talk about the whole initiation of heartbeat, which is leaky in terms of, I think it's calcium ions, or is it sodium ions? Well, it's leaky in terms of some ions, and when the ions have leaked out, and if you think of it, it's like a nerve impulse, that it's about ions moving over a membrane. And this little area that starts off the contraction leaks ions until they get to a point where it triggers like an action potential. And that then spreads through the heart, and it all then has to go back to normal. And then it leaks again until it triggers an action potential. It's not an action potential, it's a contraction of the heart, but it does it on its own without the need for a nerve impulse. The wall of the heart we describe with a particular word. It's the myocardium, made of cardiac muscle, which is myogenic. Alright, so this idea of not needing a nerve impulse. Now the heart does have nerves attached to it to speed it up and slow it down, but it doesn't need a nerve impulse to actually initiate contraction. Okay, so that's important. It's just as well, actually, because nerves, of course, don't actually heal very well, do they? They don't repair. If you think about it, if somebody needed a heart transplant and you required a nerve to make the heart work, how are you going to achieve that? So, you take the old heart out, you put a new one in, you stitch up a nerve. Well, it's not going to work straight away, is it? So, the fact that it doesn't need a nerve to actually make it work is very useful. And you don't really ever get, I don't think you ever get the connection of a nerve with a transplanted heart. The body relies on other things like changes in carbon dioxide levels, temperature, hormones like adrenaline, that sort of thing, to change the heart rate. So that's one useful thing. One side is thicker than the other. Which side? Oh. It's the left side, so the right side of the heart. This side is thicker. This is my heart. Right hand, left hand. It's the left side of the heart is the thicker. Yeah? I know. The right side of the diagram, but it's the left side of the heart. And the reason is, that's got to pump to the body. This side's only pumping to the lungs that sit just either side of the heart, so the right side doesn't have very much trouble pumping out to that. But the left side's got to pump to the top of your head, tips of your toes, the ends of your fingers. So the left ventricle, the wall of the left ventricle, is much thicker than the right. And it develops a higher pressure in there as well, which we'll see when we look at the graphs. So left side, I should put ventricle really. So left ventricle thicker than right. It's more muscular. This will be significant in a question somewhere. So left thicker than right. Pumps to whole body rather than just lungs. And then something else one other thing to add in here before we look at the function. Now, we've obviously got some pretty hard working muscle. You know, it's, I don't know how many heartbeats there are in an average lifetime, you could work it out. But in order to contract, the heart muscle itself needs a good blood supply. Now, the left side that's taking blood from the heart, so from the lungs, is going to be well oxygenated, so that's fine. However, the wall, if you look at your models, they're not far off. Some are sort of life, you know, large life size, small life size, around about 1 centimetre thick. Now, if you want oxygen to diffuse through that, it's going to take a while. Do you remember the red jelly blocks and the acid? That's a long time ago now, isn't it? But it took, the biggest block was 1 centimetre in each direction, wasn't it? And how long did it take the acid to diffuse into that? It was about between 10 and 20 minutes, wasn't it? So you can't get oxygen diffusing from the contents of the heart into the heart muscle fast enough. So it actually has to have its own blood supply. And that's what the diagram, the upper diagram, shows, really, on the page where you've got the two diagrams. There is an additional circulation. And these vessels have particular names. What are they? They're labelled, actually. No, there's not many of them labelled when I come to look at it. On the left side of the diagram, you've got the right coronary artery. The other ones are also coronary, the ones that are spreading around on the surface. So the myocardium has its own circulation. So we have the coronary arteries and veins. So what does the word coronary mean? No, that's a trick question I ask every year. Coronary doesn't mean heart, no. Cardiac means heart. Coronary. Yeah, coronary. Right. What route do I usually take to get this out of people? Sometime in the next, probably, I don't know, 10 years, we will have a coronation in this country. Quite likely, won't we? You know, I mean, the Queen is now, what, 91, 92, isn't she? 92, I think. So what's a coronation? Prince Charles will become king, and at his coronation he will get a crown. Yes. It's just a crown, literally crown. Coronary means crown. When you get an eclipse, which we don't very often, and you get the moon covering the sun, you get a little bright bit round the outside. It's called the corona. And Corona beer has a little sunshine on it. And who does the coroner work for? The Queen, yeah. Yeah. So coronary is to do with a crown. And if you look at these blood vessels, they crown the heart, don't they? Yeah. So you've got, you know, I can draw a very rough little sort of, here's your heart. And they come out over the surface like this. So they're like a little crown heart. It's those that we're most concerned about the health of. Because the problem is they branch off the aorta pretty much as soon as it leaves the heart. And so anything that's in the blood will go into those. Now, the principle behind that really is that, well, you know, blood straight from the heart is going to have the highest level of oxygen, but it's also going to have the highest level of nutrients and the highest level of fats and the highest level of all sorts of stuff, isn't it? So the coronary arteries are the ones that are most vulnerable. And they are vulnerable to damage and blockage. And of course, if one of those gets blocked, it cuts off the blood supply to the heart and causes a myocardial infarction, which is stopping the myocardium from contracting. In other words, a heart attack. All right, so coronary vessels are very important. The coronary circulation in its own right, you know, is the most critical bit of all this, I suppose, because without that, the heart's not going to work. I have got, I think I've got an image somewhere of, I'm not sure now, of how you can image those vessels. But of course, they treat the effects of blockages in the heart various sort of ways. I mean, they used to do a lot of bypasses, coronary bypasses, where they would take a blood vessel, I'm not sure where from, I think the armpit, and use it to bypass coronary arteries that were blocked. But what do they use now? Ever heard of anyone having a stent? Yeah? A stent is where they thread a wire into the coronary artery, and then over that wire they pass like a little tubular basket. Inside that they have like a little balloon. They then inflate the balloon, it opens this little tubular basket, and that stays open and it holds the blood vessel open. And this is now a really common, you know, I know no end of people, mind you, I was probably a son of my age, but, you know, rather than bypasses, people are now having stents fitted, which is, you know, it's a real step forward, and it's not major surgery. Well, it might feel major to somebody having it done, perhaps, but, you know, it's pretty basic when you think about it, isn't it? You know, you've got a tube that's blocked, or we'll just put something inside it and open it up. Yeah. I did when I did my master's degree, I know I keep harping back to it, but I absolutely enjoyed that more than any other study I've ever done, because I was able to choose things that really, really interested me. The second of the units that I did, the first one was on drugs, and I specialised on blood pressure drugs in that one. The second one was on medical imaging, which was quite a challenge because me and physics don't get on, but you know, you can convince people you know stuff if you know how to do it. And I had to do a big piece of work at the end of the unit, a sort of final, not an exam, but they called it the examinable component, which involved producing a poster about half the size of that board, perhaps, yeah, 8/0, so that sort of size, and a review to go with it. And we chose our own subject. And I found, I've never seen it used in this country anywhere, but it's something you might want to consider when you get on to the advanced physics unit, because you can write on a sort of medical application of the electromagnetic spectrum. Does it count as that bit? Ultrasound? No, it doesn't, does it? It's not part of the EM spectrum. No, this would be something else. But intravascular ultrasound. It's absolutely fascinating. And what they do is they have little tiny ultrasound, you know, the little transducers they run over the surface of your body to look at whatever, babies and anything else, hearts and that sort of thing. They had created these tiny little wire-based ones that they could literally thread through the coronary arteries and do little circular images of the walls of the coronary vessels. And I did a whole great big poster and 6,000 words on intravascular ultrasound. But yeah, we're talking, I don't know how long ago that was now. Over 10 years ago, and I haven't heard of it actually making any progress, which was a bit sad, really. But I used to set an assignment on heart disease, coronary heart disease, but I got a bit bored with it after a while. This is the problem, you set the same assignment year after year after year, you know, the marking process just gets, oh dear, yeah, mitochondria will have to go eventually. I think I should probably be gone before they are. Anyway, that's our heart structure stuff. The next thing we need to do is to work out how this thing actually does its job. And so we're going to look at the cardiac cycle. Okay. I realise I didn't put any sort of heading for the previous one, so we probably ought to go back and put, I ain't got room, heart structure. Cardiac cycle. Now, There are two parts to the cardiac cycle. It has to relax and fill, and then it has to contract and empty. And it's very important that each of those gets enough time. So we have the contraction bit. I always used to pronounce that systole, but it's not. It is systole. And this is the contraction. This is the emptying of the heart. So the muscles contract, squeeze the blood, and the blood is forced out. This is where the valves are going to come in very important. And then we have the relaxation in between beats, which is called diastole. Dia means across or between. All right. Now think of diameter. Diameter, dia, across, meter, measure. So you measure across a circle, and it's the diameter. Diabetes was to do with the dia bit. There is to do with passing across from the blood into the kidneys and into the urine. And other dias you can find, somewhere putting a cross or something like that into. And just a complete and utter sidetrack. Any of you seen that television programme on Going Back in Time for School? You might have seen it trailered. Where they're taking a bunch of kids have volunteered over last summer, I suppose, to do one of those sort of, what was school like in, they started off in Victorian times, and moved on to Edwardian, and it started off, it was quite interesting, really taught me quite a lot. I assumed that, early on, we wouldn't have any of the girls studying science and maths, and actually they did to start with, and then it was taken away. But boys were studying Latin at one point, and they came out of this Latin, oh, no idea, I'd never want to do that, that's really pointless. Why would you ever want to study Latin? If you do science, Latin is phenomenally useful. I did 3 years of Latin at school, and the way it's helped me to understand words, I just, I can't imagine how else you would ever get it. Bits, breaking words into bits, where have they come from? And this list I've got over here, and it's worth sometimes, I think, just keeping track of a few little bits of words like that. I don't think I didn't put dia on there, did I? But just that ability to break words down. Pity nobody bothered to explain to them why it actually might be useful. Anyway, diastole, enough of my rambling, is the relaxation where the muscle stops contracting and the heart refills. So we have one followed by the other, followed by the first one, followed by the other, followed by the first one, followed by the other. But systole is not just the whole heart contracting. We have, I need to add to that, atrial systole, followed by ventricular. You can't have the whole heart contracting at once. So you've got to think about how you want the blood to be moved. The blood's going to get into the heart as it just drains back from the body and the lungs. So it gets into the atria. How is it going to get from the atria into the ventricles? Well, gravity will be helpful to a large extent. But, if you're an upright animal like we are, that's fine. What if you're a horizontal animal with a heart that needs to get the blood from the atria into the ventricles? Just think a cat or a dog or something like that. How is the blood going to get from atria to ventricles? The atria contract first. So the atria pump the blood into the ventricles through the bicuspid and tricuspid valves. Then the ventricles contract. Right? So you go top down with the atria, and then bottom up with the ventricles. And the ventricles are then pushing the blood out of the arteries. Okay? And this all starts in one particular part of the heart and follows a particular pattern due to a conduction system. It's like a sort of network of wires. It's not wires, it's fibres that run through the heart. And the conduction system is quite important. Well, it's very important. So that you get the sequence right. This has got to be a little diagram. We don't need all the blood vessels on this, so if we just have the outline of the heart like that, and need the valves on the two sides, and a bit of space so I can draw in the walls as well. So that's my rough heart for this. It's very thick at the bottom, that's not healthy. The whole process starts at this point where I said we've got leaky cells. And this comes up on the exam. All right, what role does this thing play? And it's a little group of cells here called the sinoatrial node. Node is like a little lump, if you like, or SA node for short, or even just SAN. And that's the little bunch of leaky cells where the whole thing starts. So this initiates the contraction, but it's also a point that can control the rate. So it initiates contraction and controls the rate. That's where you attach your nerves. So once that bit's depolarized, that's an important word, the wave of depolarization spreads out across the atria, so they contract downwards. So here's our wave of depolarization. Let's lay that in colour. So this is a wave of depolarization. So this results in contraction. And so that's got the top contracting down. Now that hits another little patch of tissue. And it's a little area where the atria and ventricles meet. And it's about here. Oh, that's a bit central. You won't usually find it drawn quite that specifically. But this is called the atrioventricular node. So you, or AV node. Done it a bit low, but it doesn't really matter. And that's responsible for diverting this wave of polarization, because we don't want it to just spread down the heart, do we? If you think about it. If the contraction started at the top of the heart and just squeezed down, you just end up with all the blood in the bottom of the heart. We want the top to go down, but the bottom to go up. So we actually want the next bit of contraction to start down here at the point, the apex. So what we have from here is a conduction system that runs through the septum and then up the walls of the myocardium. So we've got, these are just branching fibres. We have a little bit here called the AV bundle. This is, oh dear, it's just a little bundle of fibres, slightly out of line there. So that's just a little link, if you like, the AV bundle. But then this bit down the septum of the heart has two names associated with it, which are a little unusual, should we say. Down here, this is known as the bundle of his. His with a capital H because it's a name. And every year I go away and look it up and try and remember who his was. And every year I forget again. It's one of those things that doesn't stick in my mind. But to add to that, the bundle of his is made of a particular sort of fibres. Now, composed of... There are two ways of spelling this. The first way, I suspect, is the correct way. But I think the second way has been introduced to make English people stop being so silly with foreign words. Because the original spelling was this. So, being English, those of us who are, you look at that word, and what do you say? Purkinje. Yeah, it's not. The way they spell it now is Purkyne. So that is to make us pronounce it properly. It's Purkyne. Purkyne. I suspect it might actually be more like Purkinya or something like that. But yeah. So that's how we spell it now, isn't it? Crazy, but Purkyne tissue. Yeah, you have to change a spelling just because we won't pronounce it properly. It's interesting, isn't it? Because English is full of difficult pronunciations. Somebody posted a thing on my Facebook thing the other day, and look at it, and when you look at the different ways we pronounce the same groups of letters, it's ridiculous. And yeah, we just make this assumption, though, that in other languages we just say what we see, don't we? We don't even make an effort. However, yeah, so this bundle of his, then, is composed of Purkyne tissue, and the Purkyne tissue conducts a wave of depolarization to the apex of the heart. So the contraction can then go up from the bottom. So if I go back to my red, because that was my wave of contraction, you then get the wave of contraction is up this way. Right, so at the top, excuse my coughing, at the top here we've got the atrial systole, and then at the bottom we've got the ventricular systole. So it keeps it organised, and then the whole lot relaxes. All right, so top down, bottom up, relax. Top down, bottom up, relax. Now, there's quite a bit to do on the electrical changes, the pressure changes, the opening and closing of valves and things like that. But because we're down to 6 people out of 10, I know, I never thought we were going to be a full class today. And this is a tricky bit that's coming up, and I'm starting to lose the ability to breathe again. I'm going to leave the rest of this section on describing the pressure changes and things. If you look at the next sheets that you've got here, you've got this one with a little sequence of 4 images down the left-hand side showing how the systole and diastole form a sequence. But next to it, you've got this fairly horrific-looking set of graphs all on one panel. Over the other side, you've got the same set, it's the same image, but with a lot of labelling on it. That's what I need to do next. And I think if I try and go on to do that now, I'm only going to have to do it again anyway. So I think that's a fairly tidy place for me to stop now and to actually, be able to retain a voice for tomorrow as well. And so tomorrow I will pick up there and then go on to the blood vessels, circulation and control. And that should fit in fine. It's not a big problem at all. I mean, at the moment, obviously, you've got things to be working on anyway, in terms of assignment and that sort of thing. So, or starting to work out a revision plan. So, I think, stopping at this point is, not entirely unproductive. So, I will stop my recording at that point.